Yes, thank you very much for your kind introduction, Silke and Marcel. We are lucky to be here, and we're really looking forward to give you our insights into our research that we are conducting. And I think Simone will start with giving a small, let's say, overview on the systems and on the brain organoids, and then I will take over for a while to give you my view on the Retina organoids, and then in the end we will talk about a little bit of the current models that we have, chip systems and disease modeling. And in the end, of course, there's another poll, as Silke said, in between. And of course, feel free to write us emails if you have any specific questions in that field. Okay, Simone, you want to start?
Yes, great. Thank you so much for the invitation to talk here and for the kind introductions to the organizers and the hosts. And so I have the honor of starting introducing human brain organoids to you today. And let's see. Yes. So first of all, why are we even interested in studying the human brain specifically? Why can we not use a mouse brain? And I think you can appreciate this from this two-scale depiction of the size of the mouse and the human brain, that the human brain is much larger than the mouse brain. And in particular, the neocortex in the human is composed of more than 16 billion neurons that connect to each other at more than 160 trillion synapses. So it is this mind-boggling complexity that has allowed the complex cognitive traits of our species, but may also have come with diverse neurological and psychiatric disorders. And so this is where, at least for me, really the motivation stems from. And also, if we now want to understand these structural differences, I think we really need to look at development, because the structures that we see in adult that are quite different between the different species that you can see again depicted here happens during development. And this is when really most of the neurogenesis is happening when the neurons and also glial cell types of the adult brain are predominantly being produced. And so the question that if we want to understand human brain development, how can we actually study this? Of course, we can have systems neuroscience or psychology approaches. We can, for example, study epidemiology or use non-invasive imaging approaches. We can use animal models that lend themselves to these kinds of approaches, but also to cellular molecular biology. And of course, we can use primary human tissue, but especially if we're talking about human brain development, this is a very difficult resource to get access to, as brain development happens in utero. And also, it's very difficult to experimentally manipulate if we do get access. And so really, this is why for this field, I think the stem cell derived models, such as brain organoids, have really been a breakthrough in understanding human processes of brain development. And so here's just a very brief overview of basically reprogramming of cells into different lineages. Many of you may be familiar with this. So we can take adult differentiated cell types, such as adult fibroblast cells. We can reprogram these to induce pluripotent stem cells with these so-called Yamanaka factors, or we could also use embryonic stem cells. And if we have these pluripotent stem cells, we can then, here in green, we can then induce the differentiation to diverse lineages. And this has been done for many years for different types of cells in different tissues, as you can see here, for example, cardiomyocytes, adipocytes, different types of neurons, hematopoietic lineage, pancreatic lineage. All of this can be done in 2D, but what is important for us now is the 3D differentiation. And basically 3D differentiation can lead to the formation of so-called organoids, and these have also been developed for many of these different organ systems. But today we particularly talk about the brain organoids. You can see here the workflow. We use human-induced pluripotent stem cells that can be, as I just mentioned, derived, for example, from skin. And in the lab, we can differentiate these cultures into brain region-specific organoids within basically several months. So here is a picture from day 1 of differentiation, when we basically have a collection of cells that's just a few hundred micrometers in size, to at day 90, where we can have organoids that can be 3 to 5 millimeters in size. And what is important for us as developmental neurobiologists is that the organoids recapitulate many of the hallmark features of human brain development. So here I'm, for example, showing you an immunostaining for different cell type markers. And so I'll start here from the right. You can see SOX2, and this is a marker for neural stem cells. You can see that these form kind of an anatomical structure, some kind of rosettes. Outside of these rosettes, you can see TBR2 positive cells. These are intermediate progenitor cells that are also located basically on a layer on top of these SOX2 positive cells in the developing human brain. And eventually we have TUJ1 positive newborn neurons that are even further to the outside. And so there's some kind of layering of different cell types that's reminiscent of the developing human neocortex. And additionally, these organoids also display endogenous calcium activity. These are some recordings from my lab, but many different labs have shown this. So that these cells are also physiologically active. And so with that, it's now been possible with this kind of approach to model different brain regions even, so we can do an unguided differentiation that will lead to organoids that have many different brain regions, or we can give specific signaling molecules into our differentiation media that will guide the formation of specific brain regions. And so with that, perhaps I would like to have a short stopping point and ask Silke to make the next poll available so we can get your input as well.
Yes, I have to stop screen sharing real quick for this, but the next poll is coming up. Here we go. Okay, so answers are coming in. So maybe a few more seconds. So if you still want to type something in, feel free. And then I will quickly share the results. All right.
The answer to that question is, of course, yes. It is challenging.
Will we all see the results, Silke, or?
Yeah, you should see them.
Yeah, I think it's only possible to see for those that answered.
Oh.
So maybe I will just copy them into the chat so everybody can see them. Okay. Here we go.
Yeah. Yeah, so these are very valid points, like the size limitations, for example, that probably relate to the lack of vascularization in those organoids. So we often have this problem of like a dead core in the center of the organoid because the nutrients can't really get there. I'm assuming time maybe refers to the length of time we can culture these organoids and the processes we can recapitulate. And I think to that I would say that I think there's some consensus in the field that we can definitely model some of these main processes of proliferation and neurogenesis that happen in the second trimester of gestation. And perhaps we can go with modeling network activity all the way to the like equivalent of the third trimester of gestation. But I think beyond that, it gets even more controversial as well. And I guess this already refers to the last 2 points that were answered. Maybe we can go back to the slide, Silke, please. Another limitation on the complexity is that if we have these brain region specific organoids, we can study brain region intrinsic processes, but of course a lot of neuronal networks depend on several different brain areas. And in this, to answer the questions like this, in the last couple of years, assembloids have become popular, and these are basically organoids where you generate 2 different types of organoids independently first, and then you fuse them, as you can see in this image. So this is one of the current developments to make the organoids a more comprehensive model, alongside with other developments that, for example, try to generate vascularization, try to include additional cell types such as microglia cells that come from a different lineage and are normally not around in the standard organoid protocols, at least in the early stages. And so these are some of the current developments that are going on to further improve the human brain organoid models. And so with this, I would like to end my brief overview of human brain organoids with an overview of the applications of them to answer different questions. So the first one, of course, from a developmental neurobiologist point of view with a clinical eye, brain organoids have been used to study congenital brain malformations, and they can structurally recapitulate to some extent what is going on. So for example, they have been used to understand how the Zika virus that emerged in 2015 and induced that was then inducing microcephaly, mostly in Brazil, how the microcephaly is caused and which kind of cell types are affected. Just as an example, but also, of course, brain organoids have been used to understand how genes that, when mutated, cause microcephaly are acting at the cellular level. The second major point here is about psychiatric disorders, where we often have a functional maturation problem, but often it also goes back to problems with neurodevelopment. And so there are studies that use, for example, iPSC derived from patients that have maybe schizophrenia or autism spectrum disorders to understand the processes that are malfunctional at the cellular and molecular level. Similarly, organoids are being used to study neurodegenerative diseases and also to induce aging phenotypes. A fourth big interest is to use organoids for compound screening and automated readouts. So this is also, I think, where the field is still developing into a lot. And there's a whole neurotoxicity perspective to this. And of course, the pharmaceutical industry has a lot of interest in that direction. And finally, if you think back to my first slide, where I started with human evolution and the differences in brain size between our species and other species, and despite of the similarity of the genomes, there's such a big difference in brain size and function, and using organoids, we can now have access to studying the cellular and molecular processes that may have contributed to the different brain size and function between the different species. And so with that, I would like to hand over to Stefan Liebau.
Yes, thank you very much, Simone. Also, you spared me some of the basic introduction of things in organoids. And we are interested in organoids of the neurosensory system, of the human neurosensory system. And of course, as you see in this slide, the problem for us is also that the human retina, so the optic apparatus that conducts then the light information to the brain, is highly complex. And one would say, okay, it's so complex that it's maybe unreachable to at least mimic in vitro only the most basic parts of this organ. And if you consider, okay, I have to see if I can change it. Yes, perfect. And if you consider that you want to, let's say, model organogenesis like Simone in the brain and cellular interactions, or if you want to model a disease, do drug screenings, toxicities screens, and of course think about therapies like cell replacement or cell regeneration, then you might say, okay, where are you starting here in vitro? And I mean, for other systems in the, let's say, endodermal system for gut and other organs, animal models might be pretty close to the human system, but not in the neural system. And I would not say that the human eye is superior compared to other animals. It's not more complicated. It's not better. It's not having more subtypes of cells. But the answer is simple. None of the animals represents in any way the human eye. Either it does have different photoreceptors with other chromatic view, or it's a completely different one, like in the fly, or it's just not reachable. I mean, you cannot always take the eyes of a monkey, and of course, in regular current cell cultures with, let's say, one type of cell, you can also not model in any way, in any kind, that complicated computing of light that is happening in the retina. I just want to last a few words on our induced pluripotent stem cell system. So a couple of years ago, we chose a very easy system of not taking human skin fibroblasts, but the keratinocytes from human plaque hair. For us, it was extremely helpful because we had also the plan to take care for disease modeling from patients of really young age. And you can imagine that a skin biopsy is hurtful and it has to be done in the clinics. And so we decided we use that plaque hair system, and we found also that you can transport hair at room temperature in a normal buffer. So it's a pretty nice system to generate somatic cells for the reprogramming into human-induced pluripotent stem cells. But nevertheless, get back to the retinal organoids, and if we do so, you can see here one in situ. So that's a phase contrast picture of a retinal organoid. The size varies usually from 1/4 to 1 millimeter in size, also depending, of course, on the age. And it's pretty interesting that you can culture these retinal organoids up to 2 years now in culture. It doesn't mean that they always get much better, but if you look at the photoreceptors, for example, that are located then on the outside of that organoid, they get really more mature by time. I said that the lamination of the human eye is really complicated, and lucky for us, biology knows that, and also developmental biology. So if you take cells from the retinal field from your cultures and you leave them alone in a 3-dimensional room, in media, then the elimination is done by biology and by life. So after a while, around 80 to 100 days, you see elimination. So every little part of the retina, every cell type is laminated in the correct position, and all the cell types of the retina are there. So all the neurons and the non-neuronal cells. That makes it really, really nice to not only look at the morphology of these organoids, but also looking at comparable function and ultrastructural level. And talking about function, it's not only that you can stain for the different cell types of these organoids, but you can also stain for chemicals, for molecules that are used in the human retina to either conduct light or to develop cells. And we chose here some stainings for the photoreceptors for arrestin 3, and then you can see that there's a specific sodium potassium ATPase, and also a specific marker for the outer segment of the photoreceptors that are, of course, the crucial part of the photoreceptor to really conduct and sense the light and give it further to other neurons. That's what I said. On ultrastructure level, we also looked at parts of the outside of the photoreceptor. So you can see that they at least construct also inner segments, connecting to the outer segments. Of course, if you look at these pictures here, you would say, well, it doesn't really look like a human retina, and you're absolutely right. So the most problematic part of the retinal organoid is exactly this outer segment. You have to imagine it's on the outside of a round ball, and that ball is swimming in media, and every step of working with such an organoid, or preparing it, or cutting it, or embedding it, gives a whole lot of sharing forces to the outside of these balls. And on the outside are the outer segments that are really fragile. So that is some of the problem that we face, is that the outer segments, which are really crucial and really important, often get washed away or destroyed during processing. Nevertheless, in the more inner parts, where you would see and find the synapses between different cells, so for example, between the photoreceptors, the horizontal cells, and the bipolar cells, here in the lower right picture, you can see that what we find in our cultures is really representing in every step and every way ultrastructurally what we would also find in the physiological human retina. So what we did in using this model is, of course, what we have as a prerequisite is the function. Also, as Simone said, that we conducted calcium imaging together with Vadut from the Institute for Ophthalmic Research here in Tübingen, and he also found and showed us that not only the photoreceptors are able to sense the light, but they also are able to give further the information to the other neurons of the neural retina, so the bipolar cells and the ganglion cells. So these retinal organoids are really looking into their surrounding. So one of the first studies that we conducted here in cooperation also with the industry is that they asked us, okay, you have now a human model, which we don't have. And for us, it's most interesting to see whether the plant therapies that we have might be able to mimic in your model system. Because on the long hand, we want to know, can we use a human pluripotent stem cell-based retinal organoid model as something for preclinical studies? I mean, we are here in the 3R Center webinar, and that means also that all the models that we have should at least, as close as possibly, mimic the human physiological system to be able to really use that model to replace, reduce, refine other animal models. And they asked us, okay, we have different subtypes of viruses that should be trophic for certain cells in the retina, and they already had a therapeutic approach on the line of the NIH, and so they asked us, okay, can you recapitulate what we see in other animals? And so they gave us different subtypes of viruses, of AAVs, and said, okay, they should be able to go into the photoreceptors, for example. And in the end, we said, no, it doesn't. So in the human system, it seems that a different subtype of the virus would have been perfect for your approach, and that has been proved also in other studies. So it's really important, this human model, to recapitulate therapeutic approaches in that case. Okay, so first of all, a little summary. So using these retinal organoids, and this is a nice scanning electron microscopy picture here, represents more or less a full human model. So all the cells are there, all the lamination is there, the function is there. Of course, it's not a perfect retina, but maybe we'll get a bit further into that, and we will also talk about the chip systems later on. It's really flexible, it's really tailorable, so we can use reporter systems and other things to make it even better for the questions that we are asking. And for us, it's also really important that we have a system that is really reliable. So every time that we make these retinal organoids, it's a real retinal organoid. It's not a different region or something. So this system is really perfect for asking questions of development and disease. Okay, Simone, I think your part is coming again. If you dismute, we can hear you.
Sorry. Yes. Now, hopefully you can hear me. So now that you've heard some broad introductions from me and Stefan about brain and retinal organoids, we wanted to share with you some of the projects that are going on in our labs as real examples of how we are using these models to address our research questions. And so here I'm giving an overview into what's at the core of my research interest. And you can appreciate from this Time magazine cover how the first 9 months shape the rest of your life. That it seems like even in the popular press, it's appreciated now that there may be a lot of processes during pregnancy that are shaped by the environment. Or in other words, there may be a lot of plasticity actually in development that we actually don't understand at the cellular and molecular level so far. And so in order to address this, we have a series of projects where we're interested in understanding how different environmental impacts, such as maternal infection, maternal stress, toxins that the embryo may be exposed to through the mother, also pharmaceuticals that the mother might have to ingest to control for other disorders. All of these things could pass the maternal fetal barriers, such as the placenta, and lead to changes in brain development that will then later on have an effect, for example, increasing the risk for neurodevelopmental disorders. And so what we are using the organoids for is that we're aiming to understand the molecular mechanisms of this prenatal programming in a human context. In a second pillar of research, we are interested in genetic neurodevelopmental disorders. And in a final pillar, we are also interested in model system development. And so here's just to give a bit more context on this first pillar, where we're looking at different maternal factors. So we recently reviewed a large body of literature in this area that has used organoids to model these. So we can model viral infections. I already alluded to the Zika virus before. Also maternal stress, substance abuse and medication have an effect. Fetal hypoxia is also really important in having an effect on brain development. For example, hypoxia induced by a placenta insufficiency, or maybe also for prematurely born babies. We know that hypoxia has severe consequences. And so if all of these adversities can cross these physiological barriers, such as the maternal liver, the placenta and the fetal blood-brain interfaces, so basically the early stage blood-brain barrier, these can lead to major neurogenic defects such as cell death, defects in the proliferation of neural stem cells, and also defects in differentiation of neural stem cells to the different types of neuron. And what we are particularly interested in the lab is how then these cellular defects could lead to this increased risk for neurodevelopmental disorders such as autism spectrum disorders that have been shown epidemiologically. And we are particularly interested in maternal immune activation, because a lot of these different environmental adversities that you can see here on the left actually might act through maternal immune activation as a common pathway. And so this is one of the projects we are focusing on in the lab. Then just to give you a flavor of another project of what we're doing with these brain organoids in the area of genetic neurodevelopmental disorders, that we're collaborating with the pediatrics department here on a very rare neurogenetic disorder, Pontocerebellar hypoplasia. You can appreciate from this scanning here that in this 2-month-old baby, there's a big black hole here, and the cerebellum is actually extremely small. And so this is a classical manifestation of pontocerebellar hypoplasia. And this disorder is often diagnosed within the first couple of weeks of birth. And from a biologist's point of view, it's very interesting because one subtype of this disease, actually the most common one, is caused by a specific point mutation in CEN54. And CEN54 is part of a complex called the CEN complex, which is involved in splicing introns out of tRNAs. So pre-tRNAs, some pre-tRNAs have introns that need to be spliced out in order to generate a mature tRNA, which is used for protein translation. And this is defective probably when there's this point mutation CEN54 and leads to this very severe but also very brain region specific phenotype. And so there's nothing known on the molecular pathology, on the mechanism of disease, and we are aiming to reveal this now using the organoids and patient-derived induced pluripotent stem cells. And this work is also interesting because we are not only collaborating with the clinicians and the stem cell experts here at the Hertie Institute, but also with PCH Familie, which is a parents' initiative, and they are really active in this area and are really helping to push this project further. And finally, of course, you alluded to it before yourself, the organoids are not a perfect model system yet, and there's always room for improvement. So we are working on further developing this model system together with Peter Loskill here in Tübingen, and we are aiming to make a Neocortex-on-chip. So a microfluidic chip that will allow us to generate brain organoids in a much more controlled manner. And here are just some of our very preliminary stainings where we did a differentiation into neocortical fate on a chip as a proof of concept that this is working. And yeah, so this is thanks to the collaboration with Peter Loskill and Ning Zhang in his lab. And we would like to use such a chip in the future for both developmental neurobiology, basic research questions, but I think it will also be particularly helpful for neurotoxicity questions. And with that, I'm playing the ball back to Stefan.
Absolutely, thank you. Okay, so we're now in the chip system, and so I will also tell you a little bit about our chip that we also have with Peter Loskill, of course. He's our chipologist here, and what I did not mention, why we constructed this chip is, as I said, okay, the retinal organoids are already brilliant and reliable, and they have everything they must have. there's one big thing that, besides the vascularization, that they don't have, and that is the crucial interaction between the retinal pigment epithelium and the photoreceptors, or the rest of the neuroretina. And the retinal pigment epithelium is that lamina that makes your eye, your background, black. So if you look someone deeply in the eye, it will still be black in the background. And most of that non-existing color is done by the absorption of light in the RPE. And the problem in the organoid field, in the retinal organoid field, is that you do not have this interaction between the RPE cells and the photoreceptors. And there's so many crucial functions between these two guys. So the RPE is nutrifying the photoreceptors. It's trafficking the outer segments. It's building a really crucial barrier for everything. So that interaction is one of the most important interactions between two completely different cell types in the retina. And I don't think that you can see my mouse here, right? I can only explain by color. So if you look at the chip, so there's on that little magnification on the left upper side, you can see that the photoreceptors, which have these, say, greenish and turquoise color, they are embedded in these fingertips of the RPE cells, these dark yellow ones, and below these RPE, there's a membrane, a porous membrane, and below that we also have that fluidics, because the outer retinal nutrifying with blood is coming from below the RPE. The intraretinal blood vessels is something that we also are trying to model at the moment. So we are differentiating also from pluripotent stem cells the different cell types that we need for vessels, like the endothelial cells, smooth muscle cells, satellite cells, etc. But of course, we have not found that perfect system of really bringing vessels into the retinal organ. But nevertheless, this chip already gave us a really high advance in that field, because we are now able to mimic that interaction between these two cell types, and we also able to mimic this barrier, this outer blood retinal barrier, which is mostly performed by the RPE cells. And if you look at the lamination on the left side, and then you're on the right side, you see that a photograph of someone holding one of these chips. So you have 4 little chambers where the retinal organoids are placed perfectly above these retinal pigment epithelial cells. And also very important is that you have a timeline, a workflow, where we first of all see these RPE cells, and then after a while, when they really gain that black color, so it takes a while until they really differentiate and build a perfect 2D layer, and then we put these retinal organoids on top, and then the interaction can start. And after we were successful in constructing this chip and got our first pictures, for us was also important, which you see in the upper left corner in the fluorescence staining, that not only there's a perfect distance between the photoreceptor and the RPE, which can be then elongated from the photoreceptor. So once the outer segment is running down, it can touch the RPE and then fulfill the cell-cell interaction. And what we also saw is that the RPE cells, which should traffic the outer segment, so they always phagocytosing these parts of the photoreceptors and helping to renew them, that we saw that our RPE layer is able to really take up these outer segments from the photoreceptors from the organoid. And probably it's a bit too small here, the ultrastructural level, and as I said, there's so many shearing forces in the normal media chamber. And we also were able to see that the outer segments had a really better ability to grow, and also the number of outer segments was much higher in the chip. And for the patent and also for the first publication, we also did some toxicology screens. So we took pharmacological substances which were known to have side effects on the retina, and we put them into the chip, and then we saw completely what was expected, that some of the pharmacons were barriered by the RP, so it did not affect the photoreceptors, and others which were known to go through the RPE to the photoreceptors also affected the retinal organoid, and especially the photoreceptors. Next, of course, we also tried to build a small chip, which we call the outer blood retinal barrier chip. So it's only constructed of the RPE and an endothelial layer, which is of course also really important to understand the barrier function of that OBRB, and we hope that we can improve that chip and maybe combine these chips. And this is of course something that we also want to conduct in the future, that we combine the retinal organoid chip plus the outer retinal barrier plus the inner retinal barrier, and of course all that together with vascularization, which is really something in the complete field of organoids. Everybody is trying to vascularize organoids independently of if it's a gut organoid or brain organoid or liver organoid or pancreatic organoid. The vessels are really, really needed, and this is I think one of the biggest challenges that we have at the moment in the whole organoid field. Okay, Simone told you something about clinical or disease modeling, so I will also get you something on what we're doing in disease modeling. So we have patient-specific iPS cells from people that suffer from blindness, that is based on a mutation of the so-called CRAMS protein, which is involved in polarity of cells and other stuff. So people with that mutation really get blind really early on. So it's normally an early onset disease. And now one of the work groups is trying to find the mechanism which is behind all this. And we hope that we will be able to find that in the retinal organoids. And we already have some, let's say, findings that might be responsible for having these patients getting blind in the end. So sometimes, and hopefully that we will also be able to help people in the future by understanding pathomechanisms and by understanding certain factors in disease. So we hope that we are successful here. Okay, I think, Simone, we come to an end here. I, of course, want to thank the people that did all the work, and I have the fun sitting here and let's say, giving everything to the public, and people in the lab are working 24/7 to get all the data. So thanks very much, all the guys, and also, of course, to our fundings. I have not listed everything that's at the moment. I mean, it's not that we have billions and trillions, but I think the DFG and BMBF and the medical faculty do at least a very good job in helping us performing these things.
Yeah, then I would also, of course, end by thanking my team and all our funders. And thank you very much for your attention. And I think both Stefan and I are really looking forward to your questions.
So then I take over again briefly to moderate the discussion. Thanks a lot first to the fantastic presentation, a nice tandem back and forth. And I hope we convinced everyone that this is not science fiction anymore, that this has gone into application and it will benefit patients. So I'm just looking, I'm not sure whether I have the right chat. I don't see questions at the moment. So please remember, you can just put the questions into the chat. But perhaps as a warm-up, I'm starting for some understanding. So we work with neuronal systems in a similar way, but not in the retina and also not the cortex. So I see this retina and I try to understand what is outside and inside. I mean, normally the photoreceptors look towards the brain, like the other way than one would think. So how is it in these cultures? Do they look to the medium or to the plastic?
It's easy to remember. So the outer retina is the outer retina, both in the organoid and also in the organism. So although the photoreceptors are looking in towards the brain, they are still the outer part of the retina. It's probably something that one has to really get used to, but in our retinal organoids, the photoreceptors look at the medium, so they look outside more or less.
Okay, outside is always a definition. So they look up, they look at you.
Yes, yes, a returning glance from the dish.
Okay, so that would mean you could put the retinal pigment epithelial cells on top there.
Yes.
And if you want to do it in the chip, you have to turn it around.
Yeah, in the chip, only one part of the organoid is touching the RPE. So if the RPE is yes, camera, the RPE is below, and then you put the organoid on top, then you would see also a difference between those parts of the organoid that are not touching the RPE and those parts of the organoid that are touching the RPE. So it's still round. We're trying to make it flat and all those things, but at the moment we have just the RPE below. And then we just place the organoid on top, and the lower part of the organoid is touching it.
And then, of course, probably the key question everybody was thinking about. So part of the retina is the optic nerve. So do you have any type of an exit there, or are there ideas how you could generate this?
Yes and yes. Okay, let's get one step. Let's say one step back. The problem is same as Simone said, that the core of organoids, the bigger they grow, the worse the core gets. And this is also the case in retinal organoids. So the ganglion cells that build up the optic nerve are to a certain degree dying after time. And now we are trying to give them a chance to project into another organoid, for example, a brain organoid. So we need to let's say open that organoid, so that in the middle we have, let's say here, in the middle we have here the ganglion cells, and they should then try to be able to conduct in the other direction to so-called assembloid. And we are also trying to construct a chip with two different chambers where the ganglion cells then will be able to build a kind of an optic nerve, also giving them oligodendrocytes for myelinization and stuff. So, but this is at the moment future talk, so we don't have it yet.
Okay, and that would lead to the electrophysiology and getting light-induced electrical signals and so on. Cool. I got another question coming in for Simone, I would say. You had mentioned it very briefly in the slide, that was about the barrier function towards the organoids, and whether there's chances to model the barrier function, or whether you would think the organoids just show their function without the barriers, or are these different research fields?
So at the moment, as we are using the organoids, we don't have the barriers, but there are, I think now like as this field is expanding dramatically at the moment, there are increasingly publications that have models for these barriers. For example, I think last summer was a publication from the Lancaster lab where they generated choroid plexus, and they were able to generate some kind of blood-brain barrier. And I guess I'm not too deep into this field, but one could imagine that one could also generate placental barriers. And I guess in the end, I have the impression that this whole field tries to kind of get like get add more and more layers of complexity by combining these different organoids together in assembloids, and maybe even in the very long distance future, like connecting different organs together on chip or somehow in vitro to understand communication between different organs. I mean, I think in neuroscience now, for example, there's a lot of interest in gut-brain interaction, and I could imagine that in the future there could be probably there's already ongoing developments where you can combine kind of gut and brain organoids to see how interaction can happen.
Okay.
I just want to mention that there's a couple of questions now in the Q&A.
Yes. So let's pick up perhaps the assembloids. So this is kind of the next step. So in the organoid fields, a lot of people try to connect organoids with blood vessels. I think the record is 12 or 15 of them. So liver, kidney, lung, and so on. But then in the brain, it would be brain regions, and you wouldn't have a blood supply in between. But of course, the question is, how big can you make an assembloid without getting the necrotic core? I guess this is one of the questions.
Yeah, I guess it's still very much an active field of development. I don't know what to say to some extent. I think it's really being developed, and we will see what the future brings.
And the most important thing is, even if you have vessels in an organoid, they must be perfusible. So it's not enough only to see some vessel-like structures, but they really must be able to conduct notification or notifying, and they must be able to, let's say, conduct media and also other cells. I mean, same with if you want to induce an immune cells in your organoids, also there you need sometimes vessels to give them a natural way to get into the tissue they need. And I think also Robert Mertens asked, yes, we do have vascular organoids, and we also have regionalized vascular cells for the retina. Like, if you want, I can give you a link to PubMed where we published something about the vessel cells. And I think the assembloid thing is here also a question: how many different organoids can you assemble? I think this it's only of course of a matter if you combine things that also need and want to be combined. But the number of how many different ones, and we have, for example, we now have also regionalized thalamic organoids, and we try to assemble them with retinal organoids to make up the first stage of the optic nerve. We'll see how far that goes. I mean,
yeah. What is fun about these seminars, you know, once people get triggered, the fantasy can just go on. So one kind of obvious question with a good fantasy is, can you use that also for brain tumor research? Is there something on gliomas and organoids?
Yes, definitely. So there are definitely, it's an active area that I didn't touch on, but there is definitely several models for tumors in organoids, studying either different mutation or introducing also patient material into organoids to study how they behave. And yeah, it's a big area.
I think the cancer organoid field is completely different. Cancer organoids are sometimes hard to understand because nobody really knows how many different kinds of cells there are in such an organoid. Is it always only the end point of a cancer cell, or do you also get the cancer initiating cells, so the cancer stem cells? But this field is really growing, and it's really interesting. And I think although I do not do any oncogenesis, I think that's a big hope for the future, that in vitro systems are now being able to do toxicity screens or even pharmacological screens. So, for example, if you have a pancreas cancer, you can already take organoids from this cancer and test different therapy regimens to see whether it might help the patient.
Good. So in all exciting discussions, the time is running away. I know like different people have planned different things afterwards, so we have to cut the discussion. I'm sure that our presenters will be happy to answer emails or anything if questions are coming up. That's a nice way to continue this. I hope we got an interest into the field and showed that there are alternatives to the animal approach, which have the advantage of being human, and they are applicable to some extent. So thanks a lot for all the discussion input. Thanks to the presenter, and a very nice evening. And I hope we see you again for the next seminar. Goodbye.
So we have to thank you for the organization.
Thank you, everybody. And don't forget to take the quiz. Is in the chat, the link.
Thank you. Bye-bye.
Bye-bye.
Bye-bye.